everyone. Now we will discuss briefly about the physiology of metabolism related to diabetes management. As we all know, diabetes is a chronic metabolic disease. This section is very important to understand the basic disturbances in carbohydrate, fat, and protein metabolism in diabetic conditions. Based on this knowledge, we can properly guide our patients regarding the nutritional factors involved in the diabetes management, and also we can guide them about the types of exercise and dietary modification during exercise. Furthermore, we can better understand the pathophysiology of acute metabolic emergencies like diabetic ketoacidosis, hyperosmolar states, and lactic acidosis and hypoglycemia, and the mode of action of many current and future drugs. So let's start. So Sir Hans Krebs, discoverer of the citric acid cycle, he has described 3 stages in the oxidation of food to produce energy. So in the first stage, large molecules in the food are broken down into the smaller unit that play a central role in the metabolism. So it's shown here, carbohydrates are hydrolyzed to simple sugars like glucose, fats are hydrolyzed to glycerol and fatty acids, proteins are hydrolyzed to amino acids, and no useful energy is generated in this stage. So in the second stage, most of the first stage products like glucose, fatty acids, glycerol, and several amino acids are further metabolized and converted into acetyl coenzyme. Some ATPs are generated in this stage, but the amount is very small compared with that obtained from the complete oxidation of the acetyl unit of the acetyl coenzyme. So metabolism of each molecules include 2 processes, catabolism and anabolism. So first we will discuss catabolism of carbohydrate. So glucose produced in the first stage further metabolized to pyruvate and lactate in the mammalian cells by the pathway of glycolysis. Glycolysis occurs in 2 ways, in the presence of oxygen, in the absence of oxygen. So we will discuss in brief about each process. So glycolysis in the presence of oxygen, we call it aerobic glycolysis. Tissues that can utilize oxygen are able to metabolize glucose into pyruvate and then pyruvate to acetyl coenzyme as an end product. A simplified schematic diagram of aerobic glycolysis is shown in this slide. As shown in this figure, glycolysis occurs in the cytoplasm at a rate largely determined by 3 unidirectional reactions catalyzed by the hexokinase enzyme, 6-phosphofructokinase enzyme, and pyruvate kinase enzyme. Of these, phosphofructokinase enzyme is the most important. So these are the rate limiting steps, and pyruvate is finally metabolized via TCA cycle that we will discuss. Now a little bit about anaerobic glycolysis. Anaerobic glycolysis can occur in the absence of oxygen when the end product of the metabolism is lactate. So normally some of the pyruvate is also converted into lactate by enzyme lactate dehydrogenase, and this reaction is reversible, as you can see here. Means LDH catalyzes the reversible formation or reduction of pyruvate into lactate and removal of lactate, that means reversible oxidation of lactate into the pyruvate in all the cells. Now, normal blood lactic acid is maintained by balance between lactic acid production and clearance or removal. So pyruvate is the basic substrate or metabolite for lactate production, and pyruvate is derived mainly from 2 sources. First source is from proteolysis that constitute around 15% by a variety of dehydrogenation and transamination reaction. So I'm not going into much detail. I'm just mentioning only important points. And a second source is very important, that is from anaerobic glycolysis we have just discussed. So this anaerobic glycolysis is the major source during hypoxic condition. TCA cycle is inhibited and the glycolysis pathway is activated to compensate for a lack of ATP production. In such situation, glucose in the cytoplasm is converted to pyruvate through a series of classic catalytic reactions. So pyruvate does not enter in the mitochondria for final oxidation, but is directly reduced to lactate in a process largely dependent on the enzyme lactate dehydrogenase enzyme. Now, source of lactate production. Glycolysis per se does not produce lactic acid, but provides its conjugate base like lactate, ATPs, and water. Hydrolysis of ATP in turn generate protons in an amount equivalent to that of lactate. It has been estimated that under basal condition, whole body lactate production is 0.8 millimole per kg per hour or 1400 millimole per 70 kg per 24 hours. Diffusion of 1400 millimole of acid into the ECF destroys 1400 millimole of bicarbonate, converting the alkali to sodium lactate. Hepatic oxidation of extracted sodium lactate regenerates lost bicarbonate in this cyclical process. So the production of lactate as fuel increases when the demand for oxygen in ATP exceeds the cellular supply, such as during strenuous exercise and infection. Major sites of lactate production include skin, erythrocytes, brain, skeletal muscle, and intestinal mucosa. So lactate removal. In the healthy, rested, overnight fasted individual, lactate production is balanced by lactate removal. Accumulation of lactate in the human body is more dangerous than the accumulation of any other molecular fuels, and a rise in serum lactate can lead to lactic acidosis. Therefore, lactate needs to be rapidly and metabolically removed from the tissues as well as from the circulation. The catabolism of lactate in cells occurs through three pathways as shown in the slide: through the oxidative phosphorylation, through the non-oxidative metabolism of lactate, and through via the renal excretion. So we will discuss in brief. When oxygen is available, irreversible lactate removal is achieved by pyruvate dehydrogenase, which converts, we have already discussed, again repeating, pyruvate into the acetyl coenzyme in the cytoplasm, and then acetyl coenzyme enters in the TCA cycle, and we call it the oxidative phosphorylation, leads to generation of 38 moles of ATP compared to 2 moles of ATP that is generated in the process of anaerobic glycolysis. So when there is a plentiful supply of oxygen to the tissues, the metabolism of pyruvate is primarily oxidative rather than reductive. Consumption of lactate by oxidative reaction occurs in all cells except erythrocytes, which lacks mitochondria. Now, systemic balance between glycolysis and PDS flux may be a key determinant of lactate levels. Now, when oxygen is not available, the other major routes for lactate removal is by non-oxidative metabolism of lactate in liver and kidneys by biosynthetic mechanism that involves synthesis of glucose from pyruvate, also known as gluconeogenesis. Quantitatively, the most important non-oxidative process of lactate removal is by gluconeogenesis by the liver and to a lesser extent by the kidneys. So the liver accounts for 50% and kidneys accounts for the 30% of the whole body lactate uptake in the resting state. Native kidneys have a major roles in lactate metabolism. Renal cortex appears to be major lactate consuming organ in the body after the liver. During resting or fasting state, gluconeogenesis assumes the major route. Glucose formed by this process is released into the circulation and it is taken by the peripheral tissues. Lactate released by these tissues in turn serves as a substrate for hepatic and renal glucose synthesis or gluconeogenesis. So the cyclical metabolic process involving the synthesis of glucose in the liver from lactate produced by anaerobic glycolysis in the muscle, and reuse of glucose by muscle for energy production is known as Cori's cycle. This operation of Cori's cycle that involves the continual production and removal of hydrogen ion is crucial for maintaining the acid-base equilibrium. Protons liberated in the cytoplasm by the hydrolysis of ATP during glycolysis are used by the mitochondrial respiratory chain for synthesis of new ATP available to drive such undergoing processes such as gluconeogenesis. So most of the lactate removal by kidneys are via gluconeogenesis that we have discussed in the previous slide rather than excretion. But when the renal threshold for lactate is reached, excretion becomes another mechanism for lactate removal. Under conditions of marked hyperlactinemia, such excretion can account for approximately 10 to 12% of renal lactate disposal. Since 30% of lactate from circulation is removed by renal gluconeogenesis as well as renal excretion, as we have already discussed, nephrectomy results in an approximately 30% decrease in exogenous lactate removal. While acidosis significantly depresses hepatic uptake of lactate, acidosis enhances renal lactate metabolism. Such adaptation takes place over 2 to 4 hours and occurs despite a reduction in renal blood flow. The renal contribution to lactate removal thus increases from 16% at pH of 7.45 to 44% at a pH of 6.75. So these changes will probably be important in human acidosis as they compensate for approximately 50% of hepatic loss of lactate metabolism. Lactate removed by kidneys are increased by acidosis and hypoglycemia. Kidney remains an effective lactate removing organs even during endotoxemia shock. They only fail during gross reduction in renal blood flow. The artificial kidneys also has a profound effect on lactate balance. So if lactate buffered fluids are used in patients who require continuous hemofiltration and who have pre-treatment hyperlactinemia, the serum lactate levels can significantly increase. In such cases, this increase can result in exacerbation of lactic acidosis. So if bicarbonate buffered replacement fluids are used, a significant correction of acidosis or acidemia can be achieved. So clinician needs to be aware of these renal effects on lactate level to understand the pathogenesis of hyperlactinemia in critically ill patients and to avoid misinterpretations and unnecessary or inappropriate diagnostic or therapeutic activities. So lactate uptake by liver and kidneys is concentration dependent and is saturable, and both organs can respond to hyperlactinemia by increasing lactate extraction by several folds above the basal rates. Because of balance between production and removal, basal lactate concentration usually ranges between 0.75 to 1 millimole. Balance between production and removal is important for optimal lactate level. Hyperlactinemia or lactic acidosis will result when its production exceeds its use or disposal. And hepatic and renal functional adequacy is of paramount importance in lactate homeostasis. Failure of these two organs, either singly or jointly, associated with anaerobic conditions like hypoxia, septicemia, will definitely lead to life-threatening lactic acidosis. Anabolism includes glycogen synthesis in liver and muscle, protein synthesis, lipogenesis, pyruvate, and intermediate of citric acid cycles are used in steroid synthesis in the body and glucose synthesis from non-carbohydrate substances like gluconeogenesis. Now, in a brief, we will discuss about the lipid metabolism. So, lipid metabolism is concerned mainly with the fatty acid and cholesterol metabolism. So, we will discuss about the fatty acid metabolism. The source of long-chain fatty acid is either dietary lipid or de novo synthesis for from acetyl coenzyme derived from carbohydrate, means lipogenesis we have just discussed. In tissues, fatty acid may be either oxidized to acetyl coenzyme by beta oxidation or um, by catabolism, or esterified to acylglycerols, means synthesis of fat, or anabolism. Triacylglycerol fat constitute body's main calorie reserve. Acetyl coenzyme formed by beta oxidation of fatty acids has several important fates like energy production via beta oxidation and TCA cycle, lipogenesis, for example, synthesis of cholesterol, and subsequently other steroids, and ketone production of ketone bodies in the liver. So in liver is a acetyl coenzyme formed from carbohydrate and fatty acid metabolism forms ketone bodies, or we call it ketogenesis, which serves as an alternative source of tissue fuel, particularly when energy is required under emergency conditions like starvation. They also serve as fuel for muscle and heart to spare carbohydrates. So in normal individual, ketone is produced in the controlled way. So from all these discussion, we can see that glucose is the main source of energy utilization, and ketone bodies serves as an alternative source of energy utilization to a certain extent. Source of ketones are fatty acid oxidation and protein breakdown. Hence, anything that leads to increased fatty acid oxidation, increase protein breakdown, or decrease glucose availability. For example, decrease insulin hormones and decrease counter-regulatory hormones will lead to ketogenesis. Now we will discuss about the metabolism of proteins. So we have discussed that in the first stage, proteins are hydrolyzed into 20 kinds of amino acids. So protein metabolism is related with the metabolism of amino acid. So a little bit about the structure of the protein. When we see the structure of amino acid, an amino acid is consist of an amino group and a carboxyl group and a unique side chain connected to a central carbon atom. So the peptide bond is formed between the carboxyl group of one amino acid and the amino group of the adjacent amino acid. Each polypeptide chain of any length has at one end an N-terminal amino acid containing free amino group, and at the other end C-terminal amino acid containing a free carboxyl group. Amino acid is a polypeptide chain and are numbered from the N-terminal end. Now, metabolism of amino acid. We have discussed that in the first stage, proteins are hydrolyzed to 20 kinds of amino acid, and metabolism of protein consists of anabolism and catabolism. So anabolism includes protein synthesis as listed here. So proteins are responsible for all bodily and cellular function as listed here. So I'm not going to read it. Now catabolism of protein is related with the catabolism of either amino group or carboxyl group. So after deamination, excess amino nitrogen is removed as urea, and carbon skeleton that remains after transamination undergoes in the following reaction: oxidized to CO2 via citric acid cycle and forms glucose via gluconeogenesis pathway and forms ketone bodies. Excess proteins breakdown is also responsible for increased ketone production. In addition to their requirement for protein synthesis, the amino acids are also the precursor of many other important compounds like urine synthesis and hormones such as adrenaline and thyroxine. Now the third stage constitute the TCA cycle, also called as Krebs cycle and oxidative phosphorylation. So acetyl coenzyme produced by carbohydrate, fat and protein metabolism brings the acetyl unit into the cycle where they are completely oxidized to carbon dioxide and H2O. So four pairs of electrons are transferred to an NAD plus and FAD for each acetyl group that is oxidized. Then ATP is generated as electrons flow from reduced form of these carriers to O2 in a process called oxidative phosphorylation. Most of the ATPs generated by degradation of foodstuffs are formed in the third stage. So summary of all the metabolism of carbohydrate, fat and protein is given in this slide. We will discuss in brief about metabolic patterns of human organs. Humans have a constant requirement for energy, but they eat only intermittently. To cope with this problem, we generally ingest food over the immediate caloric need of our vital organs and store the extra calories mainly in the form of glycogen, triglycerides, and to a lesser extent, muscle protein. In turn, during starvation, in response to various stresses, these fuel reservoirs break down to provide energy for organ metabolism and function. The two principal circulating fuel in humans are glucose and free fatty acid, which are stored intracellularly as glycogen in the liver and muscle and as triglyceride in the adipose tissue. Body protein is not, strictly speaking, a fuel reservoir. Protein molecules serve specific roles in the maintaining organ structure and function and are less expendable than glycogen or triglyceride. On the other hand, a portion of body protein is degraded during starvation and other periods of stress and provide an amino acid substrate for gluconeogenesis. Now, metabolic pattern of brain. The brain lacks fuel stores. That means it stores almost no energy as glycogen or fat and hence requires a continuous supply of glucose as a fuel, which enters freely at all the time. It consumes about 110 gram of glucose daily. The brain accounts for some 60% of utilization of glucose by the whole body in the resting state. It utilizes glucose as a fuel derived from liver, either directly from glycogen or indirectly via gluconeogenesis. The brain, unlike other organs, does not utilize free fatty acid as a fuel directly. However, during prolonged starvation, it can use energy derived from free fatty acid after its conversion to ketone bodies. The shift in fuel uses from glucose to ketone bodies is crucial in minimizing protein breakdown during starvation. So glucose is virtually the sole fuel for the human brain except during prolonged starvation. Now we will discuss in brief about metabolic pattern of muscle. The major fuel for muscle are glucose, fatty acids, and ketone bodies. Muscle differs from the brain in having a large store of glycogen. 3/4 of all the glycogen in the body is stored in the muscle. Glycogen is readily converted into glucose 6-phosphate for use within the muscle cells as it lacks the enzyme glucose 6-phosphatase that readily converts glucose 6-phosphate into glucose. So unlike the liver, it cannot export glucose, means muscle cannot give brain to glucose as a fuel. Glucose is rather retained as its preferred fuel for burst of activity, like increased muscular activity. So this enzyme is important for readily availability of glucose to other tissues. So muscle lacks this enzyme, but liver has this enzyme. Now, metabolic pattern of actively contracting muscle and resting muscle. The rate of glycolysis far exceeds that of the citric acid cycle in the actively contracting muscle. Much of the pyruvate formed under these conditions is reduced to lactate, which flows to the liver, where it is converted to glucose through Cori's cycle we have discussed. This is a shift of metabolic burden of muscle to the liver. Also, a large amount of alanine, that is a protein, is formed in active muscle by transamination of pyruvate. So, alanine, like lactate, can be converted into glucose by the liver. So, and we call it gluconeogenesis. Now, the metabolic pattern of resting muscle is quite different. In resting muscle, fatty acids are the major fuel. Ketone bodies can also serve as fuel for heart muscle, where acetoacetate is preferred to the glucose. Typical daily fuel requirement of different vital organs are given in this table. Now, metabolic pattern of kidneys. Under physiologic circumstances, the kidneys play a significant role in glucose homeostasis via gluconeogenesis, reabsorption of glucose from the glomerular filtrate, and uptake of glucose from the circulation to satisfy its own metabolic needs. Now, role of gluconeogenesis. An estimated 20 to 25% of fasting glucose or glucose release into the circulation during the fasting state or post-absorptive state originate from the gluconeogenesis in the kidneys. Interestingly, renal gluconeogenesis increased in the postprandial state and accounts for approximately 60% of endogenous glucose release during this time, which has been hypothesized as a function to facilitate repletion of glycogen stores in the liver. Reabsorption of filtered glucose. Reabsorption of filtered glucose in the urine, approximately 160 to 180 gram per day filtered glucose is reabsorbed from the glomerular filtrate in the proximal convoluted tubule via the sodium glucose co-transporter type 2 or the SGLT2 co-transporter. The remaining glucose reabsorption from the glomerular filtrate is achieved by SGLT1 transporter in the straight segment of the descending limb of the proximal tubule. SGLT2 and SGLT1 work together to reabsorb virtually all glucose filtered through the glomeruli. So under normal condition, approximately 180 gram of glucose are filtered by the glomeruli on a daily basis. Of this filtered glucose, essentially all of it's reabsorbed by SGLT2 and SGLT1, with SGLT2 reabsorbing the majority of the filtered glucose, approximately 90%. So based on this function, new drugs, SGLT2 inhibitors, are developed and being marketed. Now, metabolic changes in the diabetic state. Post-absorptive or fasting gluconeogenesis is increased in kidneys, notably gluconeogenesis by the kidneys comparable with that of the liver. Glomerular filtration of the glucose is increased and more than 180 milligram are filtered. The amount of glucose filtered into the urine increases in a linear manner and increasing plasma glucose concentration. As an adaptive response to this increase, there is an amplified expression of SGLT2 co-transporters, resulting in a raised set point for urinary glucose secretion. So through these changes, kidney tackle problem of hypoglycemia. Finally, the interplay between these tissues get altered and adjusted in various adverse situations. So this is the end of lecture. In the next lecture, we will discuss about the hormonal regulation of metabolism.